So in this video, we look at how our text annotations and the pipeline behind it, how they actually help you with reading a paper on the IUCr pages. So here we are on the landing page. Now let's copy in a link to a publication. So this is the publication we're going to look at, just a random pick. It has 3 structures in it, 3s6e, 2lq5 and 5cxt. So let's go to the publication text. So if you're not familiar with it, IUCr has a few annotations they provide to you. With going on this button here, you can find the list and you can toggle them on and off. So we are interested in the ones for the residue annotations. And if we go and we find some annotations like so, and we toggle this button on and off, you can see how they appear and disappear. So in our example here, we are now currently looking at a point mutation. As we set in our publication text, we provide annotations for point mutations and for residues, individual residues. So if we click on that, you can see we've got a list of residues that we found. So here for the structure 5cxt, so the residue was found in a number of different chains. And we have a second structure down below, and that is 3s6e. So you remember they were mentioned at the top. So these are the 2 structures where we find the residue in. If you're also eagle-eyed, you will spot that in 5cxt, we have a tyrosine in 468, which will therefore represent the mutant structure. Whereas if we go down to 3s6e, we have the asparagine, which is the wild type structure. Now let's open that one here. The residue tyrosine 468 in chain A of structure 5cxt. Right, so as you can see, we've got here our tyrosine, nicely highlighted, and it is grounded or linked to its reference in UniProt, which is the accession code Q8VH51, and the numbering from UniProt is also an asparagine. Sorry, no, it's the asparagine 468. So this is the wild type sequence in the reference for UniProt. Not the tyrosine, because the tyrosine, as we've established, is the mutant. So if you move it around and you look at it, you can also see it is provided with electron density. So we have experimental data for this tyrosine, and we've got some basic modeling statistics that tells you how well the residue fits into the data that's coming along with it. If we go down and we open the other structure, so the first thing you will notice, there are 2 entries for this residue 468, so asparagine 468 in chain A, and that is because it has been modeled in 2 different conformations, a conformation A and a conformation B, and for each of those we'll get the modeling statistics against the data, so we've got data available as well. So if we open that one, and you can indeed see that this native asparagine is available in 2 different conformations, so conformation A and a conformation B. It is again linked to its UniProt accession for reference, which is Q8VH51. So what we can establish therefore is that 5cxt and 3s6e contain the same protein based on the UniProt reference. In 5cxt we have a tyrosine mutant, whereas in 3s6e it's the wild type. So this is looking at a mutant structure, at a point mutation as we annotated it in the text and what we can get from it. So if we go further down our list, you can see we get a number of other residues that have been annotated. So here, let's have a look. Where is it gone? let's have a look at this one here. For example, the tryptophan 495. If you go and click on this one, as with the point mutation, you will see we'll find it for two different PDBe entries, so S3, S6E and 5CXT, but as we've seen from the point mutation example, it's actually the same protein considering the UniProt reference. So if we click on that one, we get this tryptophan up here, and you can see it nicely modeled into its electron density. And if we get the same, indeed, this time we have the same residues. There's no mutation involved. So if we get the same one here for 3s6e, again, you can see how it is modeled into the electron density, and we'll have some modeling statistics with it. Right, now the interesting thing about 5CXT is that it comes with a smaller peptide, which is not so obvious if you only look at one of the protein chains, which we've done so far, but we can look at another. And if we, for example, if we use the tyrosine 91, this example here, you can see that that this is actually true. So we've got 5CXT, which contains that tyrosine, and if you look carefully, the chain numbers have changed. But we can also find it in a structure 1-GMT. Now, if you remember when I introduced you to the structures of the publication earlier, there was no mentioning of 1-GMT, but we know from the PDB entry, 5CXT, that there are some other connections. So these structures are linked via a publication. And indeed, when you actually look deeper into our publication text here, you will find that this fragment here, this residue tyrosine 91 in chain B of 5CXT, is only a peptide, and it corresponds to a stretch in 1-GMT. Here we go. So now we've got those two next to each other. 1-GMT does not have experimental data, so we only see the atomic structure, but not an electron density. Whereas here we can also see the density, so gives us some evidence for modeling this peptide. And this is how these two structures are actually related. So it's via that peptide link. And again, we've got our modeling statistics here so that you get an idea of how well the structure actually fits with the data, and you can judge what the statements in this text are meaning in the context of the experimental data and the structure coordinates. In our next video, we will look at how these annotations provided by IUCr are actually used within the PDBe entry pages.